So can you see my presentation now?
We do.
Great. So thank you, Ulf, for the introduction. And as he said, my name is Johanna Sunde, and I'm a postdoc at the Linnaeus University. And I actually do a postdoc in evolutionary ecology, but I have utilized Pike as a model organisms to study processes of evolution. And my presentation today will mainly be about this, our latest population genetic study in Pike, which is about to be published in Molecular Ecology any day now. And yes, let's just move into it. Okay, so already during the first day of this presentation, there was a question concerning the population structure of Baltic Sea pike. And as was answered back then, the previous studies show somewhat contrasting results. So we don't really know for sure how it looks. So some studies suggest that there is weak genetic structuring with these large regional genetic clusters that can be found. And then other studies have showed that there is fine scale genetic structuring and that even neighboring populations in adjacent streams are differentiated from each other and are assigned to almost population specific genetic clusters. So based on this, we were really interested in finding out more. How does it actually look like? And how do different evolutionary processes shape this structuring? Yes, so first of all, we needed to find a way that we could do this to increase the knowledge from previous studies, because there are some previous studies and that they show contrasting results. It's a bit problematic and also interesting. But we needed to find a way that we could increase the knowledge. And one of the things that we thought about is that most of the previous studies have utilized, or almost all of them have utilized microsatellite markers. And in these studies, relatively few markers are used, which then limits the information that you can get from the genomic or the genomic information that you can get. And during the last decades, the next generation sequencing technologies have advanced quite a lot and the cost has gone down for these. So they now present good alternatives that can be utilized, which provide a much higher resolution on the genomic information. So we thought that we would utilize one of these methods, and then RADseq, restriction site associated DNA sequencing, seemed like an option. And it's a reduced representation method, which will provide a little bit of information throughout the genome, but you will not have all the information, but it gives a better estimate of the genome-wide variation. However, before we wanted to do this on a really large scale, we wanted to see if this was actually, if it looked like this could be a promising method for our system. So we started to do this pilot study with 3 populations that we know from previous microsatellite studies that they are differentiated and that they differ with regard to levels of differentiation. So if I see, so we have 2 populations here. Can you see my mouse? Yes. So 2 populations that are fairly similar to each other, and they spawn in wetlands close to each other. And then we have another population which is a lot more differentiated from the other populations. So we sampled DNA and performed both RAD sequencing and microsatellite genotyping. And if we have a quick look at the results, I think that the results speak for themselves. So this is the genetic structuring that we could see with microsatellites. We see that the method is actually quite good in distinguishing the population that is more differentiated than the others, but it has a problem in distinguishing between the 2 more similar populations. And RADseq, on the other hand, it did a great job in distinguishing all 3 of these populations. And this last red dot here is probably a migrator also. So it's a really good way of increasing the resolution. So based on this, we then wanted to do this on a large scale. So we sampled populations almost throughout the Baltic Sea. So from the Denmark in the south up to Umeå in the north. And we included population then from contrasting environments, and it also represented all the 3 different ecotypes. So we sampled DNA and then performed the RAD sequencing. And based on this RAD sequencing, we then analyzed both neutral and adaptive genetic variation. So if we start to look at the neutral genetic variation, this is then what we could say the newest and the most high resolution neutral genetic structuring of Baltic Sea pike. And these populations are now ordered in a latitudinal order. And in these, there are some patterns that we could see, or at least that I would like to highlight. So one of the first things that we can see is that the populations that are actually not really Baltic Sea populations, because it's the freshwater populations spawning in the mainland, they are highly differentiated from all the other populations, and they also form population-specific clusters. So this indicates that geographic isolation or isolation by resistance, which is dispersal barriers due to landscape features, is of high importance for the neutral variation. And the next pattern that I would like to highlight is this, that most of the anadromous populations were assigned to this anadromous genetic cluster. So we can see that 5 out of the 7 anadromous population that we had are assigned to the same genetic clusters, which might indicate gene flow between the populations or recent divergence. And based on previous studies indicating that there are quite low levels of gene flow among these populations, and the fact that we found in this present study that all these populations are actually significantly differentiated from each other, indicates that this recent divergence might be a more likely alternative, but this is something that we would need to study further to know for sure. Yeah, another thing that I would like to say in this context is that the levels of differentiation that we found for the populations are also higher than previous large-scale studies in the Baltic Sea, which then indicates that the populations are actually more differentiated from each other than previously believed, which then even further highlights and it supports the notion that each population should be considered a separate reproductive unit. Yes, so based on this structuring, we also wanted to analyze the contribution of different evolutionary processes, or rather modes of isolation, isolating mechanisms, how they influence the genetic structuring. And what I want you to take from this plot, it actually is a quite complex plot, but I think we can keep it as simple as possible. And then we can see that down here, all 3 all 3 explanatory variables had significant effects on the population structuring or genetic distance between individuals. So we could see that both geographic distance, environmental differences, and these landscape features or resistance significantly influenced how similar different individuals and populations are. And if we look in the plot, we can see that the direction of separation from the different explanatory variables, it points in different directions, which indicates then that there is a complex interaction between these different modes of isolation that shapes the neutral genetic variation or neutral genetic structuring in Baltic Sea pike. Yes, and one thing that I found quite cool was that we tried to do this also for only the anadromous populations, because we wanted to see how it looks like in the open system where all the populations actually have a possibility of encountering each other. and the connectivity is higher. And what we found is that it shows the exact same pattern. The values are slightly different, but the same variables are significant, and it's again differences in the direction of separation. So there seems to be a complex interaction also in the open system, which then might explain why different studies find somewhat different patterns, because it depends on how we include different samples or not. And another thing that could be pointed out here is that resistance, if we look at these values, the models including resistance had a lot higher values, which indicate that these landscape features are of a really high importance for shaping the neutral genetic variation, which is in line with what we saw in the structure plot. And then if we quickly just mention the adaptive genetic structuring. So the first thing that I think is really apparent is that there is not as clear of a structuring as in the neutral variation. And we could see some kind of potential association with latitude. There seems like there might be something there. And then we could also see that some of the populations that were neutrally differentiated all of a sudden share an adaptive genetic cluster. So for example, we have this Hamnarid and Kosta, which are both two freshwater populations, and they were highly neutrally differentiated from each other, but they share almost entirely this green adaptive genetic cluster, which indicates that they have which indicates convergent evolution, so that they have evolved adaptations to similar environmental conditions despite being reproductively separated. So it looks like latitude or some environmental similarity might be of importance. And of course, also for this, we wanted to evaluate the relative contributions of different processes or modes of isolation. And in contrast to what we saw for the neutral variation, for the adaptive variation, the environment, which is based on salinity, is the only single mode of isolation that significantly influenced the adaptive structuring. And this is not really surprising given the high importance of salinity that we know in Pike, and also the selection pressure that it imposes. But another thing that was really interesting was that, again, we see the same pattern across the structural levels. So lastly, based on the outliers that we created this adaptive data set on, we also wanted to find out, see if we could find genes under selection and see which environmental variables that might be of extra importance for Pike. So from the outlier loci, we found about 20 to 30 candidate genes. And then we searched a lot in different databases and read a lot of articles to find out the functions of all these different genes. And for many of them, we don't know what the function in fish or Pike are. However, we found some of them that are associated with osmoregulation and different ion pumps. So it seems like this salinity is really a key environmental variable for Pike, which is so consistent with previous studies and really expected. So just to summarize this really shortly, we found that all populations are differentiated, which is really important. They should be considered separate reproductive units. And then that the relative importance of different modes of isolation differed between neutral and adaptive diversity. That for neutral structuring, all of these different modes of isolation seem to have a jointly influence the structuring, whilst for adaptive similarity, environment seemed to be the main driver. And again, that salinity is really a key environmental variable of high importance for Pike. And with that, I would like to thank you all for your attention. And I would also like to thank the co-authors of the paper and a lot of different people who have helped in the field and in the lab with bioinformatic analysis and reading the manuscript and giving input on the study. Of course, also all the funders, and last but not least, all the Pikes that have been included in this study. And yes, lastly, for those of you who are interested, there is a preprint of this paper available in Authorea, so you have the link there. And yes, that's it for me. Thank you so much for your attention.